Hi, and welcome to the Origins Podcast. I'm your host, Lawrence Krauss. In this episode, I had the opportunity to talk to an old friend and colleague, Mark Mattson, who was head for a while of the former chief of the Laboratory of Neurosciences at the National Institute on Aging. I first got to know Mark a number of years ago when he and I co-organized a workshop for the Origins Project on pattern processing in the human brain. We brought together scientists from neuroscience as well as computer scientists to talk about how pattern processing is handled in the brain, and the computer scientists to talk about how pattern processing is handled there. And of course, that's an emerging field with the large language modules now that didn't exist back then. But the idea was to, among other things, to try and ensure that the mistakes of pattern processing that happen in the human brain, like schizophrenia, don't get carried over to machines. And having that discussion of neuroscientists and machine scientists was fascinating for me, and I hope for all the participants at the meeting. After that, Mark continued to work, and in fact, most recently, published a new book called Sculptor and Destroyer, Tales of Glutamate, the brain's most important neurotransmitter. Now, many of you have heard of dopamine or serotonin, other neurotransmitters. They get discussed a lot in the news, but glutamate turns out to be the chief neurotransmitter in the brain that's responsible for brain growth, synapse formation, and many of the aspects of consciousness. It's unheralded, and Mark's book discusses all of the aspects in which glutamate is important. We discussed that. We discussed his own interest, how he got involved in neurosciences, and then ultimately what it was about his early work that led him to realize the importance of glutamate. And we discussed its importance for learning, memory, synapse growth, and ultimately for understanding issues that will be relevant for AI development. It was a fascinating conversation. Mark is a serious scientist, and it was great to get back together with him and talk about this issue and about this subject that's really unheralded, but ultimately important for anyone who wants to understand how the brain works. You can watch this episode ad-free on our Substack site, Critical Mass. That site, if you subscribe to it, the donations go to support the Origins Project Foundation, the nonprofit foundation that supports this, that produces this podcast. Or you can watch it on our YouTube channel, our Origins Podcast YouTube channel, or listen to it on any site that you can listen to podcasts on. No matter how you watch it or listen to it, I'm sure you'll be fascinated and informed by this discussion about this ultimately very important subject. Well, Mark, thank you very much for agreeing to be on the podcast. It's nice to see you. It's been a while since we were together, and I forget exactly when that was, but it was a while back in Arizona.
Yeah, it was in 2015, I think. I've been following your work where you started this Origins Project initiative down there, and we're having workshops and symposia, and I was, tuning into some of them, and it was interesting. I'd had this back and forth with Francis Collins, who was the director of NIH then, about combining. Actually, what really happened is family went up to the Science Museum in New York City, and they had an exhibit where Francis Collins was like, they had a video of him going through a reel, and he was talking about, I don't know what the synergy between science and religion.
I know that. I think it was maybe the Natural History Museum, maybe.
Yeah, I think that was it.
I remember seeing that, and I was shocked to see that.
Yeah, and I got a little upset, and then I wrote an article for Frontiers in Neuroscience called "Superior Pattern Processing is the Essence of the Evolved Human Brain," and I talked about the different domains of cognition, and humans have this interesting feature of imagination, creativity, which has been important for evolution, but it can also lead you astray.
Yeah. In fact, I remember when we first start, when you talked to me, it's good that Francis Collins's misperceptions about the world got you interested in this. But I remember when we were talking about a workshop, it was pattern processing. partly because that's the key thing we understand. That I was interested, of course, in creativity and the origins of creativity, but it's a very hard topic to really get your mind around, and the knowledge is such that it's very early in that stage. But the fact that the brain is a great pattern processor was a key, and that's why we and I think you were the one that suggested it, and it influenced the last chapter of my new book, that the way to understand how something works is sometimes see when it's broken, and to understand the kind of diseases that break pattern processing, like schizophrenia, might teach you more about how the brain works, and so that was neat. And then we as you know, I like to have a public event associated with each of the workshops, and we decided to do a public event on creativity and madness, and decided that there's no better person than my old friend Johnny Depp, and we had a great public event there. You may remember.
Yeah, and back then, that's not that long ago, but since then there's been an explosion in like workshop, combining neuroscience with AI, artificial intelligence, and
Yeah, in
trying
fact,
to.
that's the other thing we did. Yeah, in fact, I think again it was you. I don't know what we decided, but that was the other thing I was going to mention. That workshop, we decided to combine it with AI and computer science people, because pattern processing, of course, is a crucial part of AI. But in particular, I think, like, once again, I think you were the one who influenced this, saying, well, if we're going to make pattern processing and eventually get maybe intelligent AI, we don't want the AI to be schizophrenic, so we should perhaps understand how it works in humans. And I thought that was to me a fascinating idea. It was a little premature at the time, that there wasn't much give and take between the two areas as I might have liked, but it was interesting to see what was known in each area. And for me, it was interesting to learn, and that's the joy. And we're going to talk about, for the most part, your new book, Sculptor and Destroyer, which sounds like a science fiction movie with Arnold Schwarzenegger or something, but it's not. It's about about glutamate, as we'll talk about, in the brain, and it's fascinating. But before we get there, and talk about brain science more generally, I've read your CV, and I know a little bit about you, but it's the gaps in that are important. I especially like to find out from scientists where they sort of got into science and how. But you were brought up on a farm, right?
Yeah, I was born in the city in Rochester, Minnesota, and my dad was the county attorney, prosecuting attorney.
Not a farmer, okay.
He didn't, no, but he grew up around horses because my grandfather trained and raced standardbred horses, trotters and pacers.
Okay.
And my father did that, and they were doing this at the county fairgrounds. They had kept their horses there. But my dad wanted to have his own training track, and he was able to buy 100 acres outside of Rochester and put it in his own training track.
Oh.
And then what we did is, for the rest of the farm, we had the neighbor farm the land, and we split the crops 50-50.
Oh, I see.
Yeah. But us kids, me and my younger brother, helped a lot with baling hay, riding on the hay rack, pulling the bales onto the rack, then going into the barn and stacking them and things like that.
Well, okay. So you were surrounded by nature in that regard. He was an attorney. I also see, I'm going to pretend like I know something, but we were talking earlier and you pointed those out to me, but behind you, those are, I think you still have some interesting horses. Those were from those, if people are wondering what they are, those are,
these are horse bits for harness horses. There's a driving bit that you put in the mouth to steer it and pull back. And then there's what's called a check bit that it goes in the upper part of their mouth, and there's a strap that keeps their head up. Because if horse is more likely to stumble if it's starting to get its head down. So you want to keep their head up.
And they don't do it automatic. They don't do it naturally. They don't keep their heads up so they don't stumble. You think the brain would figure that out, wouldn't you?
Well, some do and some don't, but you don't want to stumble in the middle of a race.
Yeah. So, okay. So getting back, your father was an attorney, but you didn't want to necessarily you didn't want to go into law or anything like that. You didn't interest you?
No, I never was really that interested, although my father would kind of talk with my mom about not a lot of details, but some things about, cases he was prosecuting. And there was one exciting day when one of the people he put in jail had escaped, and it's someone that had threatened his life, during the trial.
Oh God.
And so one day, all of a sudden, there's a bunch of sheriff's deputies show up on the farm, and they say, this guy's escaped. We think he might be
Yeah.
in the vicinity. And actually, they caught him not too far away from our farm.
Oh geez.
But
Okay.
yeah.
That'd be enough not to want to be a prosecutor, I guess.
Yeah. Yeah. So, but I was interested in nature more than anything, I think.
Now, what did your mom? What did your mom do?
Yeah, she was a nurse at St. Mary's Hospital, which is the main hospital for the Mayo Clinic, which is in Rochester.
Oh yeah, sure, in
And
Rochester, of course.
and she was there at a time. So this would be, well, around World War II, and then in the 50s in particular, when polio there was a lot of polio and kids getting polio, and the vaccine was just coming out at that time. And yeah, so she saw a lot of kids on the polio ward where she worked. Of course, it's St. Mary's Hospital, so it's a Catholic hospital. So there are a lot of nuns that she interacted with, and she'd tell interesting stories about the nuns.
Uh-huh. I assume you weren't near your parents religious, or were they, or did you?
No, not really. I think when we're young, I do remember going to church a couple times when we lived on town. After we moved out in the country, we never went to church, and but they didn't discourage it or encourage it.
Sure. Okay, well that's fine.
Yeah.
And they didn't. I mean, they didn't want you to be like a doctor or a lawyer necessarily. They didn't tell you that, be.
No, but I went to Mayo High School. It's named after the Mayo Clinic, and
Named
a
after,
lot
but
of
no connection between the two directly, just named after.
no connection. It's a public school, and a lot of the kids there were doctors' kids. In fact, one of my friends who was he was in essentially all of my AP biology, chemistry, and physics. His name is Thor Sunt.
Thor Sunt. Okay.
Thor Alf Sunt. And his father was a famous neurosurgeon at the Mayo Clinic, a pediatric neurosurgeon.
Oh, okay.
He was actually featured on, 60 Minutes way back.
Okay.
And because he got, I think it was multiple myeloma, bone cancer. And he kept doing surgeries even though he had to be like propped up.
Oh.
And but then Thor Jr., his son, he's now the chief of thoracic surgery at Mass General.
Oh, well, there you go. Like father, like son in that regard.
Yeah.
But you didn't exactly follow. You went to so the high school with having a lot of the kids of doctors, that sort of got you, I guess, sort of peer influence, encouraged in that. And did you read, by the way, did you read any science? Did you read science when you were a kid, or what got you interested in science? Was it being around the kids from other doctors, or what was it?
I think it was reading this, the biology and chemistry and the physics courses. I had my biology course. My teacher, Mr. Welty, was particularly a great teacher. And then
big difference.
But also, I would get subscriptions to Popular Science. I remember that distinctly. I would get, I got kind of the typical thing, a chemistry set.
Okay, so was that from you, or did I mean, so you were reading pop science outside of school and stuff?
That was me. That was all me. Not my parents.
From being on a farm or being around the animals, or was it?
I think so. And there's a lot of mechanics around the farm too.
Yeah, but okay. Then you but it was the biological part of sciences you wanted, not the physical sciences.
Right.
Why was that?
Well, I wasn't that great in math. Even, I did fine, A's and B's, but I don't know. I didn't really see the practical applications of like the thing, the algebra. I like geometry. I liked it a lot.
Visual, yeah, it's quite visual and innate, yeah.
Visual things, yeah.
Yeah, so it's okay. Well, and then So then you went on to Iowa State
Yeah. I
and
was in pre-vet. I was going to be a
you want to be a
veterinarian.
veterinarian.
Yeah.
That was again obviously from the animal experience.
Yeah. And back then in the early 70s, you could apply for vet school after 2 years of undergraduate work. I applied. My grade point was like 3.5, which wasn't high enough.
No.
And and then I didn't. In the ensuing 2 years, I decided not to reapply. I made a decision. I'd rather go into science.
So it was must have been a big change, a disappointment at first that you didn't get in, and then a realization that maybe you were interested in something else. It was some classes, upper level classes, or what was it?
Yeah, exactly. It was upper level classes in endocrinology and developmental biology. I was fascinated in development, from the fertilized egg on up, and how remarkable how quickly it occurs, right? It's amazing how quickly these cells are dividing and these are
Yeah,
developing.
it is amazing. Well, but it's a big emotional change, especially had you wanted to be a veterinarian from the time you were young, or
Well, it had to do with having the harness horses, right? And um, it was interesting. One summer, my dad sent me to a betting track in Ohio. It was actually in Toledo,
okay.
and and he did this purposely because he didn't want me to to train and race harness horses for a living.
Oh, I see. It would be so miserable.
Because yeah, and it's a kind of thing. If you have a lot of money to start with,
Hmm.
so it's a kind of thing. The rich get richer, and then there's these people that they can't buy the expensive yearling horses,
Hmm.
and get the good horses, and so they're kind of struggling. And in retrospect, I'm glad that he didn't encourage me to do
So
that.
you had the real world experience and decided that wasn't the life you wanted to live, but you did, but you obviously were interested enough. Did you entered university thinking you wanted to be a veterinarian?
Yes.
Yeah. I mean, I remember my own case. I remember when it was like mid level of high school somewhere. Then I grew up wanting to be a doctor. When it suddenly realization hit that I didn't want to be a doctor, and it was a shock for me. It was a bigger shock for my mother, but it took a while to get over it. A lot longer for her. But and, then decide that the science is interesting.
I had thought about going to, trying to get into med school. And by the way, it's much easier to get into med school than it is veterinary medicine school,
I've heard that. Yeah, that's
because
interesting.
there's not many veterinary medicine schools.
Okay.
But
It's
anyway,
funny. You think the
from
opposite,
the,
but
from a psychological standpoint, I just didn't like the idea of, you're doing something to a patient and then you mess up and they die.
yeah. But
And
somehow, if it's an animal, if it's a horse, it didn't seem as traumatic, or
uh, mice and rats, no. If it was my horse, it would be traumatic.
yeah, yeah. Okay. Well, okay. So upper level courses, you knew we wanted to do biology, but what drove you eventually, or maybe it wasn't initially, but what drove you towards brain science? Was that immediate, or was an evolution towards that?
No, it started with interest in endocrinology, that there's cells in one little organ, the hypothalamus at the base of your brain, or your adrenal gland, or your thyroid gland, and they produce one protein hormone, or in the case of gonads, steroid
Yeah.
hormones, and those hormones go in the blood and they can affect cells all over the body and brain. So I did a master's degree down in Texas at a small university.
I noticed you went down to Texas.
Yeah.
That's a strange jump for an
Well, it
Iowa
was
boy.
interesting. Well, as you know, so when you go to graduate school, I guess there's kind of two considerations. One is, you know, the 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 stature of the institution, but maybe as important is whose lab, who's gonna be your mentor.
Oh, absolutely. Probably more
And
important.
and so I applied a number of places, got accepted a number of places, bunch of state universities mainly. I didn't apply to Ivy League schools or anything. I didn't really, but anyway, so I got
Yeah.
in at a university in Texas, North Texas State University, and I decided to go there because there was an endocrinologist that was studying the mechanisms that control production of cortisol
Mm-hmm.
from adrenal glands. It's the stress hormone, right?
Yeah.
And and that was actually it turned out to be really good because I met my wife in Texas.
Well, that's important. Yeah.
Yeah,
She's a Texas girl?
she's a Texas girl from Waxahachie.
Oh, Waxahachie. I've been to Waxahachie myself
Oh,
many times because of the
y-
superconducting super collider when it was being built there.
oh, that's right.
Yeah.
But they never finished it.
They never finished it there, but it was a lab. They had a lab built, and I went down there, and lots of us even considered moving there. And then a lot of people did, and then they had to move somewhere else when they canceled it. But it was
Yeah.
created one of the biggest holes. It was the biggest
So
tunnel in the world for a while before they filled it in.
yeah, it was huge.
Yeah, it was going to be 40, 50
And gonna
miles
be
around.
and my wife was in the med tech program, medical technologist, and she was in work study. She was essentially washing dishes that I dirtied in the lab.
Oh,
I did
I
a
was going to
lot
say
of
something about that continuing later on, but that would be
no.
no, anyway.
So, but anyway, I did a lot of radio amino acids
Hmm.
to measure levels of cortisol,
Hmm.
and things have changed a lot. We are using glass tubes,
Hmm.
right? And we're using tritium, which is a pretty innocuous radioisotope,
Yeah,
unless you drink a bunch of it or
yeah,
something.
yeah.
And so we'd actually reuse the uh,
Oh,
the tubes,
okay.
right? And so, but she was washing it anyway. So, I talked with her and we played badminton. She's actually a good tennis player, and she was good in badminton. And then we just hit it off.
And
And
then and so she didn't mind moving back up north because I after Texas you went back to Iowa, right?
yeah, to the University of Iowa, Iowa City.
Yeah. Huh.
And
That's where you did your PhD, right?
yeah, and that was in neuroendocrinology.
So, okay, so let's see the jump. You did this hormonal activity in Texas, and both hormonal and studying, and obviously hormonal and related to your personal life. But uh, was that directing you? Well, anyway, how did that influence your decision to go into neuroendocrinology?
Yeah, I kind of applied to a number of places for PhD programs, and because I definitely didn't want to do a PhD where I was. It was,
Yeah.
and so there's a guy there, Gene Spaziani. He's passed away a few years ago now, and he was studying this fascinating neuroendocrine system that controls molting in crabs.
Okay.
So crabs have a hard shell. In order for them to grow and increase in size, they have to shed their exoskeleton.
Mm-hmm.
And there's a steroid hormone produced from cholesterol. Cortisol is produced from cholesterol. The molting hormone called ecdysone is produced from cholesterol. And the molting hormone is produced in a gland in kind of the chest of the crabs. And there's neurosecretory cells in their brain, actually in their eye stalk ganglia, that release a protein molt inhibiting hormone into the blood, and it tonically inhibits the production of the molting hormone.
Okay.
Then when the crab gets ready to molt, it usually tries to find a safe place
Mm-hmm.
because and kind of a non-stressful, safe hole under the
Yeah.
ocean, and then molt-inhibiting hormone quits being produced and the animals molt. And so we could remove the eye stalks.
Mm-hmm.
If you remove them, that will immediately cause them to molt.
Oh, really?
So we could, time exactly when we're
Yeah.
initiating the molting process. And so
One
I
of the I
studied
have
this,
to say
and
one of the many reasons I never went into biology and when is yeah the idea of removing the eye stalks or anyway go on.
and but kind of my main instead of in addition to looking at the signaling mechanisms within the glands that produce the molting hormone, the mechanisms by which MIH, molt inhibiting hormone, inhibits molting hormone production, I discovered that serotonin, a neurotransmitter that we have in our brains, in these crabs, serotonin controls the release of this molt inhibiting hormone from the neurons in the brain.
Oh, I see.
So, and then I had a...
And serotonin, just so that's the thing that a lot of people like to take when they're traveling or something.
Oh, that's melatonin.
Oh, that's melatonin. Okay, good. Just serotonin is, what is serotonin used for?
Well, antidepressants.
That's right. Serotonin,
they
antidepressants.
They increase serotonin levels.
Okay.
Yeah.
Good. Just making clear we understand that. Okay, we'll get to it. Actually, well, we'll get to various hormones and neurotransmitter stuff that in the human brain in a bit.
But that's a pretty interesting segue right there. So the most commonly used antidepressant drugs now, like Paxil, or Prozac, Paxil, etc., they act by increasing the amount of serotonin at synapses.
That's right.
And the receptors for serotonin are on neurons that use glutamate as their neurotransmitter.
Okay, well, we'll get okay. Hold that thought, because
no, I know we'll get there.
Yeah, I mean, the book's about glutamate, which is, and that's the point. You hear about dopamine, you hear about things like serotonin, all these things. When you hear about, and yet glutamate isn't something that's on people's tongue. Yet, as your book points out, and we'll talk about it, it's really essential to most, if not all, brain functioning in many, many ways. So we're almost there, though.
Listeners and viewers, keep watching.
Yeah.
Stay tuned.
Stay tuned for the big, because it's kind of amazing what you can learn about the brain and how glutamate has this really ubiquitous role in so many different ways. But I do want to focus on you for a little bit longer before we focus on the science. And so anyway, that work on the realization that serotonin, you did that when you were in North Texas?
No, that was at Iowa.
Okay, but I wonder when Iowa but you chose to go into neuroendocrinology, and I was just wondering if you already knew you sort of were interested in the brain part of it.
Yeah, because when I was in Texas, we had to study, what's controlling the production of cortisol, and it's a protein called adrenocorticotropic hormone, ACTH. It's released from neurons in the pituitary gland. And it's released in response to activation of stress circuits in your brain.
Okay.
Right. So I was really studying kind of the end readout of what happens, when you're under stress.
Yeah.
And then the other part of that is the sympathetic nervous system and epinephrine, adrenaline.
Yeah.
So anyway, that's why I get to the brain.
Okay. And so then, you did get to the brain there, and you did your PhD in neuroendocrinology, and then you went to, so you did a postdoc in neuroscience and neurobiology, and I guess that was a formative time intellectually, because that's where you made a discovery, right?
Yeah, I was fortunate. On my dissertation committee when I was at Iowa was Stan Cater, who was a very prominent neuroscientist, and for his work in studying mechanisms that control the growth of neurons and formation of synapses during development of nervous systems. But actually, he was using a very interesting system. It was snail brain neurons.
Okay.
And the reason is that individual nerve cells in the brains of these snails are they're up to about 20 times as big as nerve cells in our brains. And you can actually see the individual neurons in these snail brains when you look at them just under a dissecting, like a low magnification. You can see the individual neurons.
Wow.
And we would actually pull out the individual neurons, put them in culture, and then study their growth over time. Back then, there wasn't any digital photography, so we had to take individual 35 millimeter pictures with a 35 millimeter camera on the microscope at different time points. Then we had to go to the dark room develop the film. We had to keep track of the
Yeah.
pictures, obviously.
Yeah.
And then to quantify for example, the rate of outgrowth of an axon or a dendrite, we would have, essentially what we did is we took the photographic negative, put it in a photographic enlarger that you use for printing. So you put the image and then you trace on a piece of paper.
Oh,
This is time 1, this is time 2.
wow.
You take a ruler, and we had like a calibration thing.
Yeah. Yeah.
So it's very, very time consuming.
Yeah, people don't realize. I mean, we often complain about it. It's just amazing how much easier, so much, especially data analysis and data taking in all of sciences because of the technological changes. And I remember as a physicist, I'm a theoretical physicist for good reason, but I partly did a degree in math, so I wouldn't have to take an extra experimental physics class, but which I regret now in retrospect. But when I became chairman of the department, it was really interesting to see how, in the teaching of introductory physics labs, how my experimental colleagues, how important it was to begin to make the transition, even in the earliest classes, from the old-fashioned, writing things down in a notebook and to the new kind of data taking directly from the experiment and interface into a computer and taking the data. It's changed everything. So, and it's obviously changed our ability to understand the brain. But
and then in the early days of the computers, making sure you back everything up.
yeah, that's not just the early stages. Let me tell you,
it's still true. Yeah.
Yeah. But yeah, absolutely. But it's changed everything, and so that is, and certainly understanding brain function, the ability to be able to just do the experiments of the techniques that are now available is a huge step forward. In any case, you in those dinosaur days, you were working on your. You had a PhD. You were a new postdoc in Colorado, and you went there because of the laboratory again, or
yeah, it was. So I mentioned that Stan Cater was on my dissertation committee he had just got recruited to Colorado State University to start to head up a new program in developmental neurobiology.
Oh, I see.
And so essentially he took me with him. And then, so I started out working on these snail neurons, but then, I was more interested in humans ultimately.
Yeah.
And kind of the intermediate stage is some lower mammal, like a rat or mouse.
Yeah, somewhere between snails and us. Yeah.
So what I did is I went to a lab in what is it? SUNY Albany.
Yeah.
What's Albany?
SUNY, State University of New York in Albany. Yeah.
Yeah. And there's a lab there that had just developed methods to, essentially what you do is you get a female mouse pregnant.
Mm-hmm.
And then at a certain stage of development in the womb, 18 days in the case of the rats, you take out the embryos, and then, of course, we euthanize the mother, right? So she died a painless death, and the embryos, actually the embryos also interestingly get exposed to anesthesia.
Oh, because I wouldn't imagine, of course, yeah.
And so then we take out the brains from the embryos, and I focused on a brain region called the hippocampus. It's shaped like a seahorse. It's C-shaped. That the name hippocampus is from, I think it's Greek for seahorse or something.
Oh, okay. I wouldn't. Okay, interesting.
But anyway, so this is a brain region that's critical for learning and memory. Pretty much all the basic science, most of it on learning and memory, is focused on the hippocampus. It's very critical for spatial navigation.
Okay.
Like, so if you go out a hike in the woods, right, as you're walking along, hopefully your brain is processing where you're going, and at least that information is being encoded somewhat so you don't get lost.
Or you carry a GPS, but it's nowadays.
but this is kind of very fundamental for evolution, right?
Yeah,
is
sure. Yeah.
Navigation.
Yeah, absolutely.
And also it's where short-term memories are established and synapses in the hippocampus. And the reason is the information from all of our senses, whether it's vision, what's coming from the back of the information from your eyes goes to the back of your brain, occipital cortex, and then from there, the information converges ultimately on the hippocampus. But it's the same for your ears and your other senses. All that information is converging in time and in space as far as the neural net, the synapses on the same neurons. And it's that pairing of for example, sight and a sound at the same time. So like if you've never seen or heard a bear before, and you're walking in the woods and a bear comes out on the trail and growls at you, right? You encode that. It's a kind of a stressful experience.
Probably.
And so the next time you go out, if you hear a growling, you'll kind of, in your mind's eye, you know that's a bear. And that wouldn't happen if your hippocampus wasn't working. And that's what happens if you have Alzheimer's disease.
Oh, really? We'll get to that because
Okay.
one of the things I was going to get to it later, but we will get to it later. But I mean, for most of your career, you were chief of the Laboratory of Neuroscience at the National Institute of Aging. So obviously thinking about aging effects in humans, and obviously Alzheimer's is a big part of it. And so we'll get to there. But your book's about glutamate, and it was and your interest in glutamate began in when you were a postdoc, right? It was a discovery you made while you were a postdoc. And what was the work you were doing, and how did glutamate relate to it?
Right. So I'd take these embryonic neurons from the hippocampus and put them in the culture dish, and they settle on the surface. They're spheres. And then they attach, we have a certain molecular composition to the bottom of the dish that they like. So they'll attach, and then they start to grow several processes radially away from the sphere. The
Okay.
sphere, that's the cell body.
Yeah.
And that's where the nucleus is.
Yeah.
And so these processes will start to grow out. And then one of them, after about the first day or so in culture, one of them will start to grow faster than the others. And it will become very long. That's the axon. The other processes, they'll grow more slowly and they'll branch a lot, form a lot of branches, like the branches of a tree. Those are the dendrites. In fact, neuroscientists often use the term dendritic tree to describe, that's what they look like.
Now, I wanted to ask you a question. I didn't get it. It may be a silly question, but before we get there, I know I remember early in the book you talk, and in general, knowing that one of these things goes long. The long part is the axon. The smaller one's the dendrite. It made me wonder whether that's pre-wired in some sense, or whether it's kind of like a real forest where it's competition, and the one that wins out suddenly gets all the resources and to grow. Or and the others, whether it's predetermined which is going to become the axon, or whether that's a stochastic process,
It appears to be stochastic, and the evidence for that, we did some really cool experiments where these neurons, after a few days in culture, again, they have one process that's longer, that would become the axon. But if we cut it, if we sever it, we just take actually, these are the cell body of these neurons is like 10 micrometers, right?
Okay, so pretty small.
And the diameter of the axon is like 1 micrometer. But anyway, there may be at that point, I don't know what 50 to 100 micrometers long. So if we cut it at that point, then what will happen is one of the other processes that would have previously become a dendrite will become the axon. So the cell still has this ability to switch its polarity. So that shows that you can do some environmental manipulation and it will switch. So
you'd certainly hope it would. It makes sense to think that the brain, if that it could do that, because there could be lots of reasons. The one that's designed doesn't. So it's like, again, like a tree. If suddenly another tree gets in the way, it's important to be able to put branches out somewhere else. And Okay, so anyway, I interrupted you, but I always wondered whether that was stochastic process. So you're looking at the growth of the basically the beginning growth of a neuronal tree with axons and dendrites, and go on.
Yeah, and so my mentor and I were thinking about, and at that time in the this is like the mid '80s. At that time, the conventional wisdom was that neurotransmitters, their function is for neurons to communicate with each other at synapses after synapses have formed. But we and others had evidence that these neurons, even these embryonic neurons that are starting to grow, they do produce these neurotransmitters. So we started doing experiments. We asked, well, does, for example, glutamate, which is the major excitatory neurotransmitter in the brain, does it play a role in regulating the growth of the dendrites, the formation and maintenance of synapses? So the initial experiments were pretty simple. We just add glutamate at different concentrations to the culture medium, bathing the neurons, and then just follow them over time and see what happens. And I found that low to moderate levels of concentrations of glutamate would selectively slow down the growth of the dendrites without affecting the growth of the axon. And then I showed the reason is because the receptors for glutamate, the proteins in the membrane that mediate the response to glutamate, are located in the dendrites, but not in the axon.
Oh, I see.
And that kind of makes sense too, even from the standpoint of the adult brain, where you have these long axons and they're releasing glutamate from the presynaptic terminal. And then it's acting on the postsynaptic cell, on the dendrites.
Okay, go on. Well, I was gonna. Why don't you finish what you're gonna say, and then maybe I'll answer my question.
And then I went on to show that glutamate released from so you've got a growing axon. I have to back up a little bit. The end of each dendrite and axon, there's this highly motile structure. It's kind of like a hand.
Okay.
And it's called a growth cone.
Okay.
And it has kind of fingers that are kind of probing the environment as it moves along and grows, and kind of directing it and deciding which way to turn or whether to keep going or stop. And so the axon growth cone releases glutamate as it's growing. And as it encounters a dendrite, so this is the dendrite here, as it's releasing glutamate onto this dendrite. And then what happens is it kind of stabilizes the growth of the dendrite and allows for further interactions that lead to the establishment of a synapse that stays there and functions.
So it's okay. So yeah, when we get to the brain development, it's relevant then to establishing this synaptic structure. But I guess the question I was going to ask, and it sounded like you were saying it earlier. Originally, they thought in order for a synapse for two neurons to interact, they had to touch, right? But now, but was part of the discovery that they don't have to, that the glutamate can even if there's a gap can cause that signal transfer?
Yes, exactly. But further events have to occur that do require touching.
Yeah. Okay. Well, look, let's talk about the brain now. Let's talk about glutamate. I mean, the book is called Sculptor and Destroyer for a reason, and I think I want to read what I don't know if it's from the preface of the book or what you wrote me. I mean, it basically, you argue it's basically involved in almost all areas, in spite of the fact the public knows about things like dopamine and serotonin and everything else. That glutamate is really not only just important for development, it's also for involved brain changes, something called neuroplasticity, which we'll talk about, the energetics of the brain, and finally diseases, which are relevant for aging and other things, and it's also response to healthy lifestyles, etc. And so I want to go through those things in order, but I was kind of I thought it was kind of poetic, because you wrote me about this. You said, in a very real sense, this book was written by glutamate acting with the neuronal circuits in my brain. Glutamate played a fundamental role in generating my thoughts and the words they encode, which I then transferred to a computer keyboard. It's humbling to know that compared with the eons of evolution, the construction of neural networks during the development of my brain, the actual writing of this book seems trivial. I thought that sort of put it in perspective. That
Yeah.
mainly, and I think I'm right that your group in the '80s were the first kind of the ones to realize how potentially ubiquitous glutamate was and important in this. That it really plays a role in all aspects of neuronal networks and they also in the death of neural networks. That
Yeah.
too much it can be too much of a good thing.
Yeah.
And so, therefore, I thought it was interesting, and obviously the purpose of your book too, to use glutamate as a way of understanding how the brain works and responds to and certain issues which we want to know about more generally. And I'd like to talk to you about that, and then your own context of your work in aging, and then some other interests I know you have in healthy lifestyles. So we'll go there. Let's start with the development of the brain and glutamate's role in the development of the brain. Um, uh, and 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 so let's start. So you just pointed out that that it was it helps in the development of these ultimately the synapses by governing um, how dendrites grow and meet. But maybe you want to take us through that in a little more detail.
Yeah. So during brain development,
Actually, sorry, I realize just that I lost a note for here, but before we get there, I keep trying to think of people and put this in perspective. We talk about glutamate, but let's talk about what glutamate is before we even go into this other stuff. It's
yeah.
it's an amino acid, right? And it's one of the smallest, simplest of them, right? So why don't
Yeah,
you go into that?
yeah. So most people would know glutamate or glutamic acid as a amino acid that's a building block of proteins. There are 20 different uh, amino acids, and when the DNA encodes, essentially DNA codes for proteins.
Mm-hmm.
And amino acids are the building blocks of proteins, and glutamate's one of the building blocks. Um, it's also, and it's a simple molecule in that it has only 5 carbons. It has it's an amino acid, so the amino group means it has a NH2 nitrogen in it,
Mm-hmm.
and it also has a couple oxygens. So it has kind of the key fundamental elements of life.
Yeah.
And there's an interesting story, and I'm sure this caught your eye, Lawrence, because it has to do with cosmology and exoplanets and the possibility that there's life on other planets. But my understanding, and this was, let's see, what was her name? Her name is, I have this somewhere because I wrote it down just for, do you know Catherine Niche?
Catherine Nietzsche.
Neish. She's
She's a,
now she's at what is it? University of Western Ontario,
uh-huh.
and she works on exoplanets and
Okay.
done a lot of work on Titan.
Uh,
Titan's a moon
and the
of
interest in looking for life on Titan, which always amazes me. But anyway. Mm-hmm.
yeah, and so she did some experiments. Uh, so there's ways of measuring of detecting certain chemicals in the atmosphere of
Sure.
planets. And the way this atmosphere, and my understanding on Titan, is that there's like these molecules like drifting down towards the surface, and it's nitrogen rich
Yeah.
apparently,
Yeah.
and there's some evidence there could be water there. So now you got, I mentioned nitrogens in amino acids, hydrogen and oxygen, and then the only other thing you need is carbon, right?
Yeah.
And and so, for example, if there were CO2 or something, that is actually CO2 is the carbon source for all these molecules and all of
Yeah,
us,
obviously.
right?
And CO2 is what seems to be ubiquitous in the early evolution of the
Yeah.
of systems. But yeah, Titan has a lot of methane, liquid methane, I guess is.
Yeah. Methane could. Maybe that's what she used.
Yeah,
She
I
did
think
this.
methane is what probably.
She did this experiment where she kind of mixed these kind of potential precursors of amino acids, and then I think she did exposed to UV light, kind of like Stanley
Yeah, I was
Miller's.
just going to say, just like the
uh,
Uri Miller experiment in the
Yeah,
old days.
and she got four different amino acids, one of which was glutamate.
Yeah, because it's one of the simplest.
Yeah, it's one
And
of
I
the
mean,
simple.
and I think it's interesting because you make this statement early in the book, and I want to quote it. It turns out that genes encoding the receptors for glutamate are ancient and have been discovered in simple multicellular organisms such as slime molds and mosses. And scientists have shown that glutamate controls the growth of these primitive creatures. Um, glutamate's also been shown to control the growth of the roots of plants and mediate the plant's responses. Therefore, very early in evolution, long before there were nervous systems, glutamate came to function as an intercellular signal that controls growth and responses to environmental stimuli. So I think that the point that it's a simple amino acid, easily formed, and potentially one of the earliest ones to easily be have an abundance, probably it's not surprising in some sense that glutamate is so ubiquitous in
Yeah,
biology.
yeah, that's right. And another aspect of this has to do with energy metabolism, how
Uh-huh.
cells acquire and use energy. And in bacteria, uh, and in our cells too, glutamate plays a role in energy metabolism. And these are the kind of critical things for growth. You need energy, right?
Yeah, yeah, yeah,
And with plants, I mentioned this dendritic trees of neurons, right?
yeah.
And in many cases, these dendrites exhibit fractal geometry,
Uh-huh.
like the branches. So in the branches of a growing tree, they're essentially trying to maximize the amount of sunlight
Yeah,
that, which is energy, because they convert CO2 into glucose.
yeah.
Um, and so I think, and this is just kind of speculation with a lot of good evidence, although we do have some, that essentially what these individual neurons are doing is they're trying to maximize their access to other neurons and ultimately energy. And there's a role for the blood vessels too.
I
They
see.
have a fractal.
So being out like trees are the
They have
same
a
as trees.
fractal.
They're trying to maximize their access to the what is a scarce resource. We'll get to it. I'm still always.
Yeah,
I mean, I don't. For me, the biggest there's so many big mysteries of the brain, but one of them is energetics. The brain uses like 20 watts, and
yeah,
versus I remember when I first wrote about this. It was maybe 20 years ago, 15 years ago, in a different context, I suppose, to try and estimate what the energy usage of a electronic computer would be that would have the storage and transmission power of the brain was something like 20 terawatts, which is a million times more power. And the
yeah.
brain somehow does in 20 watts. That to me is the biggest, if you want to say quote unquote miracle, that's the most amazing aspect for me of the brain. I
It's
still,
definitely the most energy efficient information processing machine.
by orders of magnitude. By I mean,
Yeah, yeah,
it may have been computers changed so that what then was a million may only be 100 million now, or a million, but it's still a big difference between that and electronic computers.
and that energy now with the supercomputing and stuff, that's a big factor. Getting enough energy to run these.
Oh yeah, absolutely. The energy farms and
Yeah,
the environmental costs of Bitcoin and other things. Just the yeah, it's a big deal. But our brains somehow do it on a donut, which is
yeah.
or maybe not a donut for those
Well,
people.
they had
I know you're a big lifestyle
whatever
person. But
3.8 billion years, or well, not neurons per se, but they've had millions of years anyway.
yeah, or hundreds of millions maybe. But
Yeah.
yeah, no. In any case, so that is amazing. And so let's go through those topics: brain development, neuroplasticity, and then bioenergetics, and then disorders, and finally get to lifestyle. When I want to talk about your interest in lifestyle and aging and the brain in general, and then talk maybe about um um where you think the future is in terms of brain science. So in terms of brain development, we started way back when talking about the glutamate playing a role in those formation of those trees and the synapses. What else?
Yeah. Okay. So neurons are electrically excitable, like your muscle cells.
Uh-huh.
And so when glutamate binds to its receptor,
Mm-hmm.
what happens is sodium channels open in the membrane. Sodium has a positive charge.
Mm-hmm.
The sodium concentration outside of the neuron is much higher than inside the neuron. So when glutamate binds to the receptors, which is a sodium channel, it opens, the positive charge rushes in, and that's called depolarization.
Yes.
Okay. And then there's another glutamate receptor. It's a little complicated, not too bad. So you got sodium going in.
Mm-hmm.
Then glutamate also causes calcium to go in.
Mm-hmm.
Calcium's much higher concentration outside than in. And it turns out throughout evolution, calcium is a very important regulator of, for example, gene expression, determining which genes are turned on or turned off. And that means which proteins are increased or decreased. But anyway, so glutamate will stimulate the nerve cell, sodium rushing in, it will fire what's called an action potential, which quickly transmits the electrochemical signal along the axon to the next neuron.
And when I reminded me, I've always read these things, and I hadn't thought to think about it enough till I was reading your book, and then it occurred to me. So this is again a stupid question, but a question a physicist might ask. But you say that the neuron fires, but it occurred to me, is it really the opposite? I mean, so if you're depolarizing, then I'm assuming if there's polarization, there's already a potential there.
Right.
When you depolarize, you remove the potential. So I'm wondering, does the firing really mean that what you're happening is the firing is really things in reverse? If you remove the potential here, but the next neuron has a potential, then you'll get some current flow between you and the next neuron. But it'll be motivated by the fact that that the next neuron has the potential and you don't. So I'm just wondering, am I missing it? When you depolarize,
well,
is firing happening and it's like the bullet is going in the opposite direction, or no?
No, but the key thing, Lawrence, is the synapse. Okay. Which is a space between the neurons.
Oh, okay.
So the membrane isn't continuous between one neuron and another.
Okay. What I guess what I'm wondering is, so the synapse is where there's a flow right across the synapse, right?
Yeah.
And I'm just wondering if
like this.
Yeah, it's like that. And if you depolarize this one, then the current is going to flow from the one that isn't depolarized to the one that is, right? I mean, the one that isn't, the one that's still polarized to the one that isn't. No, it goes in the opposite direction.
No, the current is flowing because the ions are moving across the membrane, but there's no, the membranes of each cell are separate.
But when you say when you say it fires, I guess I want to know what do you mean when you say it fires?
That's propagation of the impulse along the membrane of an axon.
Yeah, that's what I'm wondering. And so the propagation, I mean, it's a stupid question. Maybe I'm wasting people's time. Maybe, but I would have thought if you're at this end and people have to watch instead of listen for this, but if you're at the left end and it depolarizes, I would have thought the signal would go from the part of the brain that wasn't depolarized.
Okay, what you're saying is, will it ricochet?
Yeah. Well, to some extent, ricochet. I just figure you remove a potential here, it's like a ball rolling down a hill. You have this potential because of it's polarized. You remove it here, the ball start rolling from the other end back to here. You'll still get a current, but it'll partly, it won't be like it fires because the potential is higher at the end where the glutamate was than the other end. It's the opposite. It's right. The potential's gone away here where the glutamate acted. And the signal comes from the other end of the membrane.
No, so what happens is there's energy-dependent pumps that pump the sodium back out quickly after it comes in. So it's only a transient sodium influx.
And does the firing happen after the sodium is restored?
No, it's as the sodium rushes in.
I won't waste time on it. I was just trying to picture where the actual signal comes from if you've got rid of the potential. It's called an action potential, right? Isn't it?
Yeah, yeah.
And that's
And it's initiated by opening of those sodium channels in one specific location in the membrane.
yeah. And then and okay. And I kind of, as a physicist, I think I know what's happening in my mind, and I won't bother other people. But I think but later on I'll bug you after we record, because I
Okay.
think I kind of figure what's happening in the physics of it, or the chemistry,
Okay.
depending upon what you happen. Anyway, go on.
Yeah. So, right. So, what was I going to say? So during development, glutamate's released from a growing axon onto a growing dendrite. It promotes the formation of a synapse between the presynaptic cell and the postsynaptic dendrite. And then that's fixed in place. You got the synapse. And when this neuron fires by being stimulated by a preceding neuron, and
Yeah.
then it will cause glutamate release, and then that stimulates receptors here and cause this neuron to depolarize.
Okay. Okay. So each time so the effect of nerve firing is to stimulate glutamate release.
Yes.
That's basically what it means. When you fire, you stimulate the glutamate release, which then carries the signal, if you wish, and depolarizes the next one. So the firing is really, and it really is 100% glutamate. That's the medium of this.
Yeah, and in the brain, it's that way. You can take in a stimulatory electrode and put on a neuron and stimulate it electrically. But in the brain, glutamate is the excitatory neurotransmitter. And there's a number of reasons I put in this book why I call it the most important neurotransmitter. First, more than 90% of all the neurons in the brain, the neurotransmitter they deploy is glutamate.
Mm-hmm.
The other neurotransmitters, we mentioned dopamine and serotonin. There's also GABA, which is it's kind of the brakes of the system. It's an inhibitory neurotransmitter.
Yeah.
The only way these other neurotransmitters affect activity in the brain and behavior, any behavior, emotions, learning and memory, imagination, whatever. The only way these other neurotransmitters affect those processes is by modifying the ongoing excitatory activity through these circuits of glutamate using neurons. So you can imagine you've got like this spider web of neurons that are using glutamate, and on top of that spider web, you've got these other neurons that are affecting the electrical impulses coursing through that spider web in important ways. But kind of the bottom line is, without glutamate,
you don't have any of it. But the other guys, the other ones get all the publicity, all the recognition, because they're moderating. Glutamate is the sort of the backbone of sort of the unheralded, people running the engine of the train, and it's and these other ones are more like the valves that are sort of changing what happens.
The reason glutamate is not known as well is because it's so important. Because if you drugs for glutamate are very tricky business. Drugs that affect glutamate receptors, for example, because it's critical for everything we do.
Yeah.
And if we mess with it even a little, it can screw us up.
Whereas you can mess with dopamine and serotonin and
you can mess with them a lot, and yeah.
And that's what people do, I guess, is mess with them a lot. But that's why they're more well known, is because they're the ones that people talk about, because they're the ones that pharmaceutical companies they're the ones that people are clinical, techniques and mediations are designed to affect, because you can affect things with them. That's without
psychedelics too. They're increasing serotonin at serotonin release onto glutamatergic neurons. And that's
Okay.
how they.
So okay. So now we understand that glutamate, but now we understand both why they're more well known and why glutamate is so basic. But what about what else is there any other major factor in terms of brain development that glutamate, before we get to neuroplasticity, the
Oh, yeah,
change in the
yeah,
brain,
yeah,
which I want to
yeah.
get to next, but the basic fundamental development that you want to mention?
I do. There's one other important thing. During brain development, there's a lot more neurons that are born, that is, arise from stem cells, than ultimately we end up with in the adult brain.
Okay.
So there's an overproduction of neurons, and there's kind of a pruning back. So some neurons die in the developing brain, and the ones that die are the ones that have not been able to form enduring synaptic connections.
Okay.
And what happens there, it's pretty interesting. The activity in the glutamatergic neurons will cause an increase in the production of what are called neurotrophic factors. These are proteins that promote the survival of neurons. They're also important in what we'll get to later, synaptic plasticity, changes in the function and structure of connections. And so that's kind of the second most important thing, that glutamate's not only involved in forming of synapses, it's involved in determining which cells survive.
So if the glutamate the glutamate produces the release of these what are they called,
Neurotrophic factors.
factors. So glutamate promotes their production.
Yeah, yeah.
And the ones that die don't have that production, and that's because glutamate isn't being released because there's no enduring synapse.
Yep, yep.
Okay, I think I got that. Good.
Yeah, that's exactly right. Yep.
Okay, good. Well, that's always nice. Okay, so for the formation of a healthy brain, basically glutamate is a key in all of the aspects because the brain is based on being able to create lots of synapses, and how many are there in the human brain? Hundreds of trillions or something?
About 100 trillion. There's about 100 billion nerve cells and about 1,000 times more synapses.
Yeah, so it's like if I think of cosmology as 100 billion stars, and then the brain is like 100 like 1,000 galaxies, 100 billion stars in a galaxy, 1,000 galaxies, put them together. That's about the number of synapses in a. It's interesting to think of those numbers because those numbers are so big to try and put them in perspective. And okay, so brain's developed, but the brain isn't just developed, and that's really important. And again, I think it's kind of relatively recent in my life anyway, the realization that the brain, when I was a kid, and I used to, I don't think I told you, but I wanted to be a neuroscientist. No, I didn't want to be a neuroscientist. I didn't know what neuroscience was. I wanted to be a neurosurgeon because my mother wanted me to be a doctor, and the brain is what I found most interesting from the time I was a kid. And I remember getting Psychology Today, a subscription when I was a kid, and I wanted to be a neuroscientist because I didn't know what a neurologist was. Neither my parents finished high school, but the brain was fascinating. So I have this 50-year-old awareness from all that of brain science at that time. But it seemed to me at the time that it looked like, oh, the brain develops when from in utero, and then when you're a child, and then it's fully developed, and that's it. But then of course that's not it, and the brain has great plasticity. And I kind of think that's, in that scale, relatively recent discovery about plasticity. Maybe I'm wrong.
Yeah, it is. You're right. Within that scale, it is recent. I should mention, on that time scale back then, I think it was 50s and 60s, there was a very famous neurosurgeon in Montreal, Wilder Penfield.
Wilder Penfield.
Wilder Penfield. Well,
Yeah.
So
Anyway.
So you know the story. It's he was treating patients with epileptic seizures, and back then, they still do this in some severe cases nowadays. They try to figure out where in the brain these neurons are just firing like crazy. And by the way, that's because they're releasing too much glutamate
Oh,
too
okay.
often.
There we go. We'll get to the diseases, but okay, good.
But what he did, there are no pain receptors in the brain. So you can poke around in a brain as much as you want, and the subject wouldn't feel pain. But anyway,
That's great.
but anyway, what he did,
Reminds me of an awful movie I saw. Okay, go on.
he would, trying to figure out which small piece of brain he's going to remove to try to stop the epileptic seizures, he would kind of put a stimulating electrode around different places. And these patients were not anesthetized because they didn't have to be,
Yeah,
because
because
they
there's no
don't.
pain.
Yeah. And he described how, he'd stimulate in one place and the patient would say, and he'd ask the patient, what did you experience? And they may say, well, I saw my wife coming in the room or something, or, so he could essentially, by stimulating, these people would recall sequences, not real long sequences, but nevertheless sequences of events.
Mm-hmm.
And so that was really some of the first evidence for, the brain storing specific sequences of events occurring, in a kind of like a movie.
Yeah, like a movie. Yeah. Wow. And well, I mean,
And
and that's, by the way, of course, that's what's one of the many things now that's changed with being able to new technology is you don't actually have to go in the brain so much now that we can use magnetic fields and other things we can try and image without non-invasively and see what's going on in vivo, in the sense of seeing what people are doing or thinking, or at least what processes are occurring, maybe not seeing the thoughts so much, but without actually doing what he had to do, which is go into the brain and excite it.
But as far as the neuroplasticity aspect, you're right. It's only in maybe the last 4 or 5 decades, 4 decades, let's say, this has become clear that
It seems
the
recent to me.
the brain is not hardwired.
Yeah.
It's not like an electrical circuit in this computer, or well, maybe an older computer.
Yeah.
But but it's the structure of the brain can change, and it can change, and does change in response to experience. And in fact, when we learn new things, there are subtle changes in the structure of the brain occurring. Maybe a change in the size of a few synapses, or it could be even the formation of a new synapse, or even the elimination of a synapse. And moreover, certain things we do, lifestyle factors, and we'll get into this later, like physical exercise has robust effects on structure and function of the brain. It's a great antidepressant, but it's also actually facilitates learning and memory. It will promote the formation of new synapses. It will stimulate the production of new nerve cells from stem cells, at least in the hippocampus. So these developmental mechanisms that occur so dynamically and really overtly and just remarkably during brain development, they can still occur using similar mechanisms involving glutamate, neurotrophic factors. There's some other mechanisms involved in adult brains, and moreover, we can affect those in good ways or bad ways by what we do throughout our life.
Okay, good. Yeah, that's important, and we'll focus on that at the end. I want to talk about what we can do, because I think that's what people are interested in. But right, for the right now, I want to know the mechanism. This neuroplasticity is incredibly important. The fact that it's amazing, the structure of the brain. Obviously, learning is a form of neuroplasticity because something was there that wasn't there before. Somewhere the memory is stored, and obviously we know that is real because you can retrieve it later on. So it's there. It's not ephemeral. It's in some biochemical. To the extent that all sensible people realize that everything about us is just biochemistry. That's an obvious form of neuroplasticity, but it's even broader, real structural changes. But how does glutamate specifically relate to those structural changes?
Yeah. So what's been shown, and again, looking at the hippocampus, there's been a number of Nobel Prizes given for studies of the hippocampus in various regards. One is this spatial navigation, learning and memory, how that works. And another one for these scientists discovered that by putting so say you've got two neurons in the hippocampus, and in the presynaptic neuron, glutamatergic neuron, you put a stimulating electrode. So you can stimulate that neuron however you want, different frequencies or intensities. And then there's a recording electrode in the postsynaptic neuron. So you're recording the responses of that postsynaptic neuron to the stimulation of the presynaptic neuron. And these are both glutamatergic neurons. Okay, this one group is actually up in Scandinavia. They discovered if you stimulate this presynaptic neuron at a high frequency, 100 hertz, 100 times per second, and you do that several times, and then you wait 10 minutes or you wait an hour, and then you come back and just stimulate it once, the strength of the response of the postsynaptic neuron will be stronger because you had previously stimulated at high frequency.
Okay.
So it remembers that it had been stimulated at high frequency. This is called long-term potentiation. And then that was later shown that the size of the synapse actually increases as a result of that.
I was wondering about the physical, since I'm a physicist, I was wondering about the physical mechanism for that. So stimulating a lot basically causes more dendrites to be connected or something like that, or more, and therefore
it increases
more potential sources of glutamate.
Yes. And the strengthening even of an individual synapse.
Yeah, okay.
Making it bigger.
Making it bigger, but again, maybe I'm understanding. An individual synapse involves not just two dendrites. Does it involve a bunch of them together, or just two dendrites?
No, one axon and one dendrite.
One axon and one dendrite. So there are lots of dendrites. So what I'm wondering is, if it becomes larger, does that mean there are more dendrites involved? More dendrites grow?
More synapses and more dendrites. And you can get additive effects of stimulating more than one synapse or dendrite at the same time.
Is it because, again, I'm just trying to understand the specific role of glutamate. So you excite it a lot, and it means that there's more, that somehow there's more glutamate being released because of that synaptic connection, and that is encouraging more dendrite growth, and therefore more. And so it's in that way that that glutamate is related to the plasticity of the brain by increasing that size of that synaptic connection.
Yeah. And it's more complicated than that. It involves actually elevation of calcium levels within the cells. And actually, I did a lot of work on that. Even beginning in my postdoc work, Roger Chen was a chemist at, where's he at? I think he was at Caltech or Berkeley. And he actually got a Nobel Prize. He was one of the co-developers of green fluorescent protein. But more importantly, I think, for the neuroscience field, he invented chemicals that their fluorescence changes when calcium binds to them. They're based on you may have heard of this, EGTA or EDTA. It's a calcium chelator molecule. So he modified that so that it's fluorescent and that the fluorescence changes when calcium binds. And you can introduce this into living neurons. You can image them. You can activate a synapse, and you'll see it increase in fluorescence, increase in calcium levels. And you can correlate that with strengthening of the synapse. You can even correlate it in vivo now with modern technologies using fiber optics. You can even—
I was going to say, how do you see the fluorescence? So the original fluorescence was seen, this was done in a test tube, not in the brain. It was just, you could watch it there. And then, but now you're saying you can actually put fiber optics in and see the fluorescence in vivo.
And then the key thing, so this is all showing correlations, right, between activation, release of glutamate, increase in strength of the synapse, increased calcium. That's all correlation. But that doesn't establish that glutamate is
Yeah,
necessary.
it's not causation. Correlation
Right.
is causation.
So then the other big development that came about, that's close to 25, 30 years ago, was the development of highly specific drugs that selectively block glutamate receptors without affecting any other receptor. And by using those specific blockers of glutamate receptors, it was shown that activation of glutamate receptors is essential for learning and memory. It's essential for strengthening of synapses in this particular long-term potentium. It's essential for the changes in calcium and the structural changes.
Okay. So the fact that glutamate really is an essential part of the brain, literally changing structure, and by changing structure, you mean the synaptic connections in the brain, which
Yeah.
are favored and how they're connected.
Yeah.
Okay, I get that now. Now, what about bioenergetics, which is the subject we mentioned earlier? The amazing,
Yeah.
the amazing energy efficiency of the human brain.
So all cells have energy factories, the mitochondria, which arose from, we think, bacteria-like cells, kind of a symbiotic relationship with
Yes, it's like
other cells. And
the Borg.
actually,
They
Carl
assimilated.
Sagan's first wife
Yes,
was
the woman,
and she Magoulas.
Lynn Margulis, who,
Yeah.
she was
And she,
passed away, but she was
she
amazing.
was one of the early people to have this idea, and she also thinks like out of the box, and she also had this concept of Gaia
Yeah.
that, like, well, it's we're made of billions of cells, right?
Yeah,
So each cell in our body, our brain, is part of a bigger organism.
yeah,
Could we be part of a bigger? Well, we are the Earth, right? You wrote a
yeah,
book on the climate change, right?
yeah.
So we are part of a bigger,
Yeah,
in a
she
way.
and her son got quite interested in the Gaia hypothesis. Somehow, if cells in an organism could be symbiotic, then maybe all the organisms on Earth could have a relationship. And it's the Gaia. It
And
is, in a spiritual way, it's the Gaia thing. And I kind of, but there are probably some. Go on.
You recently had a podcast with one of my good friends, Robert Sapolsky,
Yes.
right, on free will. And so, yeah, with the Gaia thing, I guess we could hypothesize that we don't have any free will, like cells in our body, and we're part of a bigger organism. And maybe,
I'm always worried about over emphasizing analogies, and I think Gaia has some nice features. To me, the biggest Gaia thing is, frankly, the fact that carbon dioxide. I wrote a book called Atom, but the fact that there is a carbon dioxide cycle in the Earth, but life dramatically changed it, because the carbon dioxide cycle on the Earth used to be 100 million years, having to do with carbon dioxide being absorbed in the ocean, being accreted in the ocean floor with calcium carbonate, and then with plate tectonics being subducted, and then being emitted 100 million years later in volcanoes, and that cycle work.
Yeah.
Life changed it from 100 million years to 100 years, cycling carbon in the atmosphere. So that's definitely an impact of life on the environment in a big way.
Yeah. Okay, but anyway, so the mitochondria are the energy factory of cells, and neurons have a lot of mitochondria, which makes sense because they, as you mentioned, they need a lot of energy. Your muscle cells, particularly if you exercise a lot, have a lot of mitochondria. And in fact, if you exercise a lot, each of your individual muscle cells will have more mitochondria than a colleague who doesn't exercise, right? So exercise will increase the ability of your muscle cells to provide energy in the form of ATP, the energy currency. Same is true with your neurons. If you use them, it will increase the number of mitochondria in the individual nerve cells.
Oh, okay. Oh, that's important to know. One of the reasons why I do this podcast, it forces me to keep using them. But anyway.
and we showed that using some modern molecular genetic mechanisms. We didn't use CRISPR, but we used another one called RNA interference, which also got a Nobel Prize. There's a certain what we call transcription factor that
Mm.
regulates a bunch of genes that are critical for the division and growth of mitochondria, a process we call mitochondrial biogenesis.
Mm-hmm.
And by manipulating that critical transcription factor, we showed that mitochondrial biogenesis, that is an increase in the number of mitochondria, was essential for these developing neurons to form synapses.
Okay.
If they don't produce more mitochondria, they'll stay alive, but they won't be able to form synapses, and their growth slows down a lot. And then, we had some evidence that aerobic exercise will also increase the number of mitochondria neurons. So, both what we're doing now, intellectually challenging,
Mm-hmm.
or turns out when we do physical exercise, a lot of neural circuits all over the brain, not just the motor neurons and the sensory motor system, but even neurons important for learning and memory, regulation of mood, anxiety, they're all activity changes.
It's interesting, yeah. Well, we'll get to the flow backwards.
Yeah.
We'll think of the flow forward now, which is how, working, how the brain, how glutamate in the brain affects the efficiency of
Right.
of energy.
So when glutamate stimulates a neuron, energy is required to restore these ion gradients across the membrane.
Uh-huh.
There's ATP dependent pumps that pump these ions back out.
Yeah.
And when you get the sodium and calcium influx, they have to work harder. And so they need more ATP. So when your neurons are active, when they're stimulated by glutamate, the mitochondria produce more ATP to provide that ATP to run these pumps that restore the gradient.
Okay, go on.
And it turns out that you mentioned these modern brain imaging techniques. There's one called functional magnetic resonance imaging, fMRI. And essentially what it's measuring is not directly measuring activity in neurons, but it's measuring blood flow.
Okay.
And just like your muscle, when your muscle cells are exercising, the blood flow to your muscle increases.
Yes.
Okay.
Mm-hmm.
The same is true with neural networks in your brain. When they're active, the blood flow increases to them. And this magnetic resonance imaging, essentially what they're measuring is protons.
It's just protons in the blood moving.
Yeah. And blood. But it's a very accurate measure of activity because there's a tight coupling of activity of neurons and blood flow. And so actually I would make the case, and I did in my book, is what these clinicians and clinical investigators, when they're doing these human studies with fMRI, what they're actually looking at is a readout of glutamatergic neuron activity, because it's glutamate that's mediating the increase in activity.
Oh, I see.
And therefore the increase in blood flow.
Increase in blood flow. Okay. And the increase in blood flow, of course, is so it's initiating the activity, and the increase in blood flow is required to deliver the ATP,
the glucose,
and
the glucose or the ketones, glucose or ketones to the neurons. Yeah. And oxygen, of course.
And exactly. And oxygen, which is required for the whole thing to work efficiently.
That's another interesting story. I think I mentioned it in my book. So oxygen, we need it to live. It's also very toxic.
Yeah, well, if oxygen was around in the environment at the beginning of the Earth, we wouldn't be here.
No.
Because it oxidizes.
So we developed ways to deal with, there's oxygen free radicals form, right?
Yeah.
It's oxidizing. And so we have antioxidant defenses. Actually, many of them. There's multiple ways that our cells protect themselves against the oxygen free radicals that are produced during the production of ATP in the mitochondria, which is essentially, you got glucose and oxygen, And just like, when you light your fire in your fireplace, you need oxygen. You need that to produce ATP. Yeah, so the increased blood flow, increased oxygen, increased energy sources, and there
Well,
you
this is a
go.
good segue because I want to talk about free radical. I want to talk about neurological disorders. Before we get there, um, as you has pointed out, I mean, and I think I described, and I think it's accurate to describe life as controlled burning. Um, we really was it is, and if there was too much oxygen early in the history of the Earth, there would have been uncontrolled burning. There wouldn't have been any energy left, and for organic materials to, and we're only there because we do controlled burning pretty effectively.
That's true.
We were going to get to you point out the free radicals and oxygen, and we're talking about controlled burning a little bit. Life is controlled burning, which is, as I say, the way I think of it. And the energetics. We kind of understand how glutamate helps with that. The producing more ATP and having more sites for ATP, which is the energy generating molecule of in all of life. This may seem a stupid question, but I can't resist asking you, especially as someone like you. I know exercises a lot, but as you point out, when you're not exercising, the brain is using much of that energy in the body. And I do find, if in my periods of intense intellectual activity, it may be because I'm eating less too, but
Hmm.
I exercise, but in terms of controlling my weight, I find that my weight also gets controlled well. If I have periods of intense intellectual activity, I seem to be burning a lot of calories and
Yeah.
and controlling my weight almost as effectively as if I'm exercising. Is that crazy?
No, it's not crazy. You can, as you said, at rest there, but you can one right now what you and I are doing,
Mm-hmm.
we're burning more brain's burning more calories than if we were just watching a movie or
Sure.
something like that. Yeah.
Yeah, and I mean, the human body is about an 80-watt, 90-watt heater when you're just at rest, and then when you're doing stuff, maybe 120 to 100 something, whatever. So, and the brain is using 20 watts, I guess, on average. I don't know. I don't know what the total. Actually, do you know the number? You must know the number, maybe. How variation of the maximum wattage when the brain is intensively working versus when it's resting, or do you know?
I don't know off the top of my
It'd be
head.
fascinating for me to know.
It's overall less at sleep, and it's most when either interesting, maybe actually physical exercise we don't know for sure because it's hard to measure
Yeah, that's true.
when you're moving, but
Yeah, it's true. It's harder to measure when you're moving, even if you have electrodes, I guess, in your head. But it'd be nice to know if it was varying by 20 watts or 50, whatever. It'd be interesting to
oh, I
know
see. I
how
see.
much, and then one could work out. But I certainly have that
But it could
experience. But
be
it's
you
not.
may be eating less. You may be
It's
eating less too. Yeah.
correlation is not causation, as we point out earlier, and it's got to remember that. So I've always been suspicious, but I have noticed in those periods of intense intellectual activity that I seem to control my weight almost as well as when I'm exercising, because I'm not always doing both. Sometimes I'm doing both, and then of course it's conflated and it's hard to tell. But before we get to health, let's get to not health. You point out free radicals and and the body has to get rid of them because they're toxic in different ways. And we hear about it all the time. I think a lot of verbiage is meaningless that's put in a lot of the media. But let's talk about glutamate and neuronal disorders and aging to some extent. How all of that combines together.
Yeah. Well, but there's an interesting story on the first evidence that glutamate can excite the brain was during World War II, there was this Japanese scientist at Keio University in Tokyo, Hayashi was his last name, and he had dogs. He drilled a hole in their skull, and he was essentially exposing the brain to different chemicals and seeing if the dogs had any responses. And then when he put a lot of glutamate on the dog's brain, it had seizures, like epileptic seizures. So that was really kind of evidence that glutamate could cause excitation. And so what happens in epileptic seizures is that, oh, then there was this incident in Canada, another very important thing. Some people they'd all eaten at the same restaurant, and shortly thereafter, within a day or two, they had seizures. And these are people who they never had a seizure before. One of them died even, the seizures were so bad. And then they also had amnesia. What's called anterograde amnesia. They couldn't remember anything new, kind of like what happens in Alzheimer's disease.
Oh, okay.
And so what happened, very interesting story. They'd all eaten shellfish. They ordered the same thing from the menu, the shellfish. And then it turned out these shellfish had high levels of a chemical called domoic acid, which selectively and with very high potency activates glutamate receptors.
Oh.
So that's what was causing their seizures. And then we actually use this in the lab. The first study, we'll get to this maybe, but our first intermittent fasting study was we found if we maintain rats on intermittent fasting and then give them this same domoic acid or another one, canic acid, they're protected against seizures. And their memory is preserved, relatively anyway. And so anyway, but then actually,
we will get to that eventually.
my first. So when I was looking at the effects way back in my postdoc of glutamate on outgrowth of dendrites and synapse formation, I discovered that a high level of glutamate would kill the neurons.
Okay, that's really important. It overexcites them or something, right?
It excites them to death. They cannot restore the ion gradients. They keep firing like crazy. Free radicals just keep being produced like crazy. The free radicals damage all the molecules in cell DNA, proteins, membranes, and they can die. And we call that excitotoxicity. So you can demonstrate that in a dish, like I found when I was a postdoc. You can demonstrate it in animals, rats, mice, dogs, and we think that's and then the drugs used to treat epilepsy are drugs that quiet down the neurons, either by, they don't really use glutamate receptor blockers much because they have more side effects with learning and memory and, having problems. But sodium channel blockers, so there's drugs that block the movement of sodium in, so it lessens the depolarization. And then there's other drugs that enhance the GABA, the inhibitory neurotransmitter. Like Valium was one, you in my generation may know about Valium, used to treat anxiety. But anyway, so neurons can be killed by too much glutamate. That's what's occurring in epileptic seizures. There's also
that's what's causing the seizure is too much glutamate, and there's just all this random firing. Okay.
Yeah. But also the death of neurons that occurs following a stroke and following a traumatic brain injury, to some extent, that involves overactivity of glutamate receptors. Because what happens is when you get damage to neurons, they spew out their glutamate. So they have a high concentration glutamate inside, it's low outside. And the only place glutamate should normally be is inside the neurons or at the synapse, but not else. But anyway, stroke, traumatic brain injury, in animal models anyway, if you give the animals drugs that block the glutamate receptors, it can lessen the amount of brain tissue that's damaged and improve the outcome. There were clinical trials in stroke of some of these drugs, but they didn't pick out an effect. These stroke trials are very hard to do because there's so much variability between individuals in which blood vessel was occluded, how long was the clot in the blood vessel, how long did it take the person to get to the hospital. Another problem and I think one of the reasons for a lot of problems in translating animal findings to humans is in our animal studies, we generally use genetically pure animals.
Yeah.
Humans are not, we have a lot of gene intermixing.
Yeah.
And so there's a lot of variability between individuals on, for example, how much brain damage they will have following a stroke. There's environmental factors that,
by the way, just so we keep clear, the stroke is just an occlusion of the blood, and therefore the energy, blood can't get the nutrients and necessary oxygen and it causes local kind of brain death, I guess.
Yep. Yep. And when the neurons continue to be excited by glutamate when they're not getting enough energy, that is not good.
Okay,
Go down.
and so the point is, the glutamate, they're still exciting it, but there's no energy, and that's
exactly.
Now, so we know that glutamate's related to these issues, these disorders, but do we know what is causing in the one case, the excess of glutamate, or as you age, or do we know what's causing that, or is that just an observed effect and we don't yet know the causes?
Well, with the stroke and traumatic brain injury, we know the cause.
Yeah, with a stroke, but
with the epileptic seizure, the ultimate cause, there's genetic factors, there's even developmental factors. Some children will have seizures when they're little, but then they disappear. And, so the answer there is we don't know exactly what's causing this imbalance between excitatory glutamatergic and the GABAergic inhibitory that leads to an excitatory imbalance that then starts to cascade out of control in a seizure. Now, I guess we could talk about aging. So with aging, everything goes wrong.
Tell me about it.
Yeah. So there's increased oxidative damage to DNA, proteins, membranes. Mitochondria don't work as well as we age. And then there's this process called autophagy, which you may have heard of.
No.
It's a kind of a cellular garbage disposal and removal system. So we have these really elegant systems when there's DNA repair mechanisms.
So the removal mechanism removes things like free radicals as well, or?
Yeah. Yep. So there's both removal of the potentially damaging things like free radicals or abnormally aggregated proteins like the amyloid in Alzheimer's, for example. And then there are repair mechanisms. DNA repair is a very elegant system and that's what prevents us from getting mutations. Our DNA is being damaged all the time by free radicals, but it's very efficiently repaired. But with aging, that repair efficiency goes down, and that's why aging is a major risk factor for cancers, because cancers are generally caused by a mutation that's not repaired.
Oh, I see.
And
I get you.
And there are other problems that occur that are more specific. There's a tendency towards an excitatory imbalance that occurs. Elderly people are more likely to get seizures than middle-aged people or young people.
Yeah.
We think in Alzheimer's disease, there's a very early excitatory imbalance. Patients with Alzheimer's disease have about a 30-fold increased likelihood of having seizures compared to someone the same age who doesn't have Alzheimer's disease.
Oh, interesting.
Okay,
now,
so problems with mitochondria means cells maybe can't generate enough ATP with aging, problems with damage and repair systems, and then problems in controlling excitability, which may be downstream of some of these other generic age-related changes.
Okay, now these are all effects and certainly relate. These are the problems. What role? What I want to, since we're talking about glutamate, in what sense is glutamate related to these particular problems?
Yeah. Okay, so in most of these disorders, epilepsy, Alzheimer's disease, stroke, it's glutamatergic neurons that die. And at least in the animal models, the neurons seem to be dying because they're being overexcited by glutamate. Okay, and then the other evidence has to do with disease specific. It has to do with.
Let me interrupt a second. Does that mean that whatever that there are inhibitory mechanisms that are not operating effectively for the
Good,
oh,
good,
okay.
good. Yes,
Okay. Thank you.
it's true.
Okay.
There's particularly this has been shown in Alzheimer's disease in particular. With epilepsy, maybe it's not quite so clear. With Alzheimer's, very early on. Okay, I have to back up just a little bit. The inhibitory neurons that use GABA as a neurotransmitter, they are much more active than the glutamatergic neurons. So they're firing normally like crazy, and their firing then inhibits the glutamatergic neurons.
Yeah. Okay.
Okay. And they are loaded with mitochondria, these inhibitory neurons. They have a high energy demand.
I see.
And they also have a high free radical exposure load. So they seem, with aging and Alzheimer's disease, they seem to be particularly vulnerable early on.
Okay, so you're not getting many mitochondria that are efficient. They're not firing as effectively, and then the inhibitory mechanism is less effective. I got you.
Yeah, the brakes are
No, the
brakes are wearing
failing.
out.
Yeah, they're wearing out.
You're going down a mountain, and the
they're yep.
The Alzheimer's cascade.
Yeah. Okay.
And then your brakes are failing.
Okay. And that's also explains why you're more exposed to seizures as you get older too. Okay. Excellent.
But I should say I've got another interesting anecdote if you have time, Lawrence.
Sure.
It has to do with Alzheimer's disease.
Mm-hmm.
We've done a lot of work trying to understand what goes wrong in Alzheimer's disease. And, others had shown that there's this amyloid protein that accumulates in the brain, forms what are called plaques, these kind of spherical deposits, and associated with the amyloid, neurons degenerate, suggesting some potential cause-effect relationship between the amyloid accumulation and the damage to the neurons. And then some of my earliest experiments after I got my first faculty position, which is actually at the University of Kentucky. I was there before I went to NIH.
Yeah, you were there for a while. Mm-hmm.
The geneticist had identified a family in which a genetic mutation in the gene that encodes for the production of this amyloid protein, that gene had a mutation. And the people who had that mutation, they would get Alzheimer's disease at an early age, in their 40s or early 50s. Okay, which is that's very unusual.
Yeah.
And it's usually genetic if it's that early. And then so the gene was sequenced, and then that means the amino acid sequence of the amyloid was established. And then everybody started, my lab was one of the first. There was a lab, Bruce Yankner at Harvard. His lab was actually the first, and then my lab, to show that this amyloid peptide self-aggregates, and when that happens on surface of neurons, it can cause them to die. But my main contribution there is I showed that even low levels of this aggregating amyloid can make neurons very vulnerable to excitotoxicity. So if you combine even low levels of the amyloid with glutamate, you can kill, so we think, at least I do, other people in the field now too, that early on in Alzheimer's disease, there's kind of this interplay between the amyloid and the control of excitability of neurons and glutamate, and you get to some tipping point and things go downhill fast.
Okay. So let's let's go from the sad to the positive. Let's look then at a positive note. Let's talk about lifestyle and the ultimate implications. First, for in the context of glutamate, for understanding how healthy lifestyles can impact on healthy brain function, and then afterwards, I do want to talk about because I know from the first time we met, I was this idea of intermittent fasting as well. So let's first just talk about lifestyle more generally. We've alluded to it already. You've alluded to already that exercise. Well, anyway, why don't you talk about how you can improve brain function and maintain
Yeah,
longevity, etc.
for the most part, what's good for the health of your cardiovascular system is good for your brain health. And that's been established in both epidemiological studies and in longitudinal, including longitudinal studies, and in even clinical trials. And so what happens in the brain with exercise is that it's very interesting. There's increased neural network activity. It's kind of the key take-home message of this is exposure of your brain cells, your muscle cells, your cardiovascular cells to a mild metabolic challenge, metabolic stress, if you will, as long as it's done intermittently and not too much intensity, is good for them. And that our cells have evolved.
What doesn't kill you makes you stronger. Is that the idea?
Yes, it's exactly right. That's something called hormesis, is another word people use to talk about this. And it's very well established with exercise and your cardiovascular and muscle. During exercise, it's a huge stress on your muscle cells.
Yeah.
And they respond in good ways they become more resistant to stress, more resilient, and have better endurance. And conversely, if you're sedentary and you overeat, it's bad for everything.
Yeah.
Including your brain. Even in children. So this is, let's start this way. Obesity is a big problem in developing countries. Started by the United States and marketed elsewhere.
Right. Yeah.
Fast food. And there's really good evidence that people with long-term obesity, type 2 diabetes, they don't think as well. Their learning and memory is poor. And this is true even, interestingly, in children. Been multiple studies showing that middle school, high school age kids that are overweight perform more poorly on various tests of learning and memory. And then this is tied into socioeconomic status too, because as you know, in the United States, there are certain regions in the southeast, where there's relatively higher levels of poverty.
Yeah.
But there's higher levels of obesity, cardiovascular disease, diabetes, stroke, many cancers. And it's because they have poor diet and lifestyle. And so I think down there, if we could get those kids, and the problem is it becomes transgenerational. Because if a kid grows up in a family that eats junk food all the time and their parents never exercise, they're not challenging themselves necessarily intellectually all the time, they're more likely to have those habits, and then it gets
passed
propagated.
On. Let me step back though. Okay, so it's clear it's I think easy for most people to realize why eating poorly and not exercising doesn't produce the challenges that help your rest of your body, your muscles and other organs function, I guess. But what's the specific impact? Why the brain? Is it do we know why? Maybe it's poor blood flow. What do we know what the sort of causatory mechanism is to poor brain function from poor lifestyle?
It's multiple mechanisms. One is actually blood vessels, because the blood vessels in your brain, just like in the ones that supply to your heart, can get atherosclerosis and so on. But there's also more direct effects on neural circuits and even other cells in the brain. Three things I can mention. One is, and I haven't mentioned yet, inflammation. Just as occurs in your blood vessels when cardiovascular disease, or in your joints with osteoarthritis, or in, a lot of cancers, actually, there's inflammation associated with it. In your brain, with aging, and much more so in Alzheimer's, Parkinson's, there's local neuroinflammation that's occurring in the brain. That is suppressed by regular exercise. Also, the cells that are involved in the inflammation in the brain, they're called microglia. They're the equivalent of macrophages in the peripheral, and they produce what are called pro-inflammatory cytokines like tumor necrosis factor, interleukin-1 beta. And so the regular exercise, by affecting signaling pathways in these microglia, can suppress their inflammatory properties.
Oh, okay. Okay. I just wondered if we knew the mechanism. Okay, that's good.
And then another mechanism is, I kind of mentioned it. So the exercise will increase production of BDNF, this neurotrophic factor that's so important for learning and memory and resilience of neurons under stressful conditions. I mentioned that. It's a very potent inducer of the gene that encodes that BDNF protein. It's very robust. If you measure in animals, and it's been looked at in cerebrospinal fluid of humans, that's the fluid that bathes the brain. It's kind of a, you can get a kind of a readout of the neurochemistry of the brain in a general sense by doing a spinal tap and measuring these things. But anyway, so that's the second one, increasing BDNF. And then a third is this increasing number of mitochondria in the neurons.
Which then, since the brain needs so much energy, therefore effectively. Okay, no, that all makes sense. It's good to know. Healthy lifestyle, healthy being fit, not just being visibly physically fit, but mentally physically fit is an important connection. Now I want to almost end, not quite, but almost end with this. So is intermittent fasting the same idea that what doesn't kill you will make you stronger? That by stressing your system, by not eating. So
in general,
I know it's a big deal for you, and I know you've written a book about it, and I know
I don't know. Did you get my book or not? Did you get
know
my
what?
book?
You never gave it to me when we were together. I didn't say anything, but
no, I didn't have it. I didn't write it till I retired.
Oh, good. Okay. Then I don't feel so bad.
It's new. It came out last year.
I know you were implementing it because you talked to me a lot about it when we were together, but
yeah. And the NIH is funding a lot of studies now on humans, intermittent fasting. But the first chapter of my book, similar to the first chapter of my Sculptor and Destroyer book, intermittent fasting is an evolutionary perspective. So during evolution, individuals whose brains and bodies functioned well in a food-deprived state had a survival advantage. That makes sense?
Sorry, go on, run that one by me again.
Individuals whose brains and bodies functioned well in a food-deprived state had a survival advantage.
Sure, of course. Yeah, that makes sense.
Yeah. Right. So during evolution, our systems, including our brain, evolved to function well in a food-deprived state.
Okay.
So when you haven't got food for a long time, you better be able to figure out how you're going to get food, use your brain, and then exert physical exertion to get the food, right?
Yeah.
So all our genetic programs and probably beneficial gene mutations, many of them had to do with improving success in getting food and therefore being able to survive and reproduce. So nowadays, we don't challenge our systems that evolved to respond to this challenge of going without food. If you eat breakfast, lunch, and dinner, and one of the key things that happens is, evolutionarily, is if you were to stop eating now, during the ensuing 10 to 12 hours, all your cells, including your brain cells, would use mainly glucose, which comes from storage in the liver. But then after 10 to 12 hours, what happens is you no longer have those glucose stores, and fats are released from your fat cells into the blood, and they're converted to what are called ketones. So this is what we call a metabolic switch from glucose to ketones. So that occurs with going without food for at least 14 to 16 hours. If you exercise in that fasted state, that enhances the elevation of ketones.
Okay.
Ketones, ketogenic diets actually are prescribed for patients with epilepsy.
Oh, okay. Interesting.
And if you go back to Roman times and some of the writings back then, when people had epileptic seizures back then, they thought they were possessed by demons.
Yeah.
Right? They're having convulsions on the ground. And they found if they lock them in a room and leave them for a day or so, the demons go away.
Okay.
So what's happening, we now know probably they aren't eating anything. They throw them in the room without any food. They aren't eating anything. Their ketones go up. Their seizures go away. The demons go away. And so with intermittent fasting, the ketones play an important role in both providing an energy source for neurons. We and others found that they enhance GABA, the inhibitory neurotransmitter, production and release onto the glutamatergic neurons.
So ketones do something glucose doesn't do, is what you're telling me.
Absolutely.
But then, but a lot of it, but there's this thing called ketosis, which is too much of it, right?
That's a clinical problem. That's extremely high ketoacidosis. Yes.
That happens if you don't eat for a long time, right?
Oh, no, you won't get that even if you don't eat for a long time.
Oh, okay.
No, it's a medical condition usually caused by problems with liver or other things.
So the idea of intermittent fasting is in some sense it enhances, ultimately your body has to turn over to ketone production rather than ketone utilization rather than glucose utilization. And ketones for some reason are good because they help with stimulate various aspects of brain function, among other things.
Yeah. And so I mentioned with epilepsy, we did a trial. We have the paper submitted, so I can't really talk about the
no worries.
results, except to say we did a trial of intermittent fasting in people at risk for cognitive impairment and Alzheimer's because of their age and metabolic status. They're between 55 and 70 years of age. They have obesity and insulin resistance, but not frank diabetes. Okay. And so we randomly assigned them to either an intermittent fasting eating pattern or kind of a regular one, breakfast, lunch, dinner. And then at baseline and after 2 months, so baseline before we started them on intermittent fasting, in 2 months, we did a battery of psychological tests focused on cognition, but also mood and other things. And then we did functional MRI. Structural MRI, we can actually measure size pretty accurately of certain brain structures. We did spinal taps and took cerebrospinal fluid. So we'll be measuring BDNF. We'll be measuring things like antioxidant things that have to do with free radical levels, inflammation markers, and so on. I can tell you that there are effects in a good way of the intermittent fasting on the brains.
Okay, and we have to wait for the rest to come out before we'll
and then there are also clinical trials of what's called a ketone ester. It's essentially the ketone that's produced during fasting. Just giving that, you can actually consume it.
Yeah.
Ingest it, and it will increase the ketone in your blood. So there are at least 2 clinical trials in Alzheimer's patients going on now.
Okay, well, that's interesting to see. We'll see what's next. In fact, that's a good way to get to near the end here. I wanted, this is, as you point out, as and the point of this was that using glutamate as an introduction allowed us to talk about a lot of fascinating aspects of the brain and your own work in it, which are fascinating for me, and I think fascinating for people in general. And I've of course learned a lot in the process. But what's the future? I mean, we talked about how when you and I first met, we talked about we wanted to run a thing on creativity and madness, but we were a little far removed from that. Pattern processing was an early stage of what's talking about, and we've talked about brain function at some basic level and understanding the processes, but we're far away from consciousness. And and but we are developing an AI. I just wanted to ask you to throw out what's the future in your mind of understanding ultimately the real questions what people want to know. I mean, obviously people want to know how to live longer and function better, but also, how to understand the real things that make us human: creativity, longing, consciousness, self-awareness, and
Well,
and then to what extent AI might play a role in any of that. So it's big question, but I'll give you a few minutes to answer.
well, that article I mentioned that I wrote, Superior Pattern Processing,
Yeah,
I put the word superior in there because I think we're only better than animals in terms of not necessarily qualitatively, but quantitatively, and maybe, you know, I often people think, we're way above other animals in so many respects. I don't think we are. I think language, the evolution of language was a huge, huge factor
yeah. Hmm.
that propelled us way above, and you can pass information down, and you can speed up things. Things like consciousness. I don't know. How do you define consciousness? I
Well,
mean,
that's
it
the
depends how
problem.
you
If you
do.
look at my book, no, I don't think anyone can very well.
I have a podcast, Lawrence. It's called Brain Ponderings, and
Yeah.
I've had a number of, but I had one on consciousness. It was with Friston, I guess, Carl Friston,
Uh-huh.
uh, somebody else. But the key thing there, we're using a human term
Yeah.
without really defining. It's like to me, it's like saying God without defining
Yeah.
what exactly do you mean? What are the particular characteristics
Yeah. Oh, look, I
of
couldn't
this?
agree with you more. I mean,
Yeah,
again, my friend Noam Chomsky was saying to me, it's maybe the wrong question calling consciousness.
yeah,
We don't
yeah,
even, it's one of the most poorly defined terms and one of the most frequently written about. But let's just talk about even, and let's being less
yeah,
ephemeral.
yeah,
What's the
yeah.
most exciting area of brain research that you think is next?
No, I think this the kind of the transition and understanding from now we have a pretty good understanding of learning and memory,
Uh-huh.
at least kind of the basics. You know, two neurons are required,
Yeah.
and you need glutamate and
Yeah.
calcium and all this. Uh, but then we know a little bit about short and long-term memory. What we don't have is this, how the brain is taking all sorts of different information, integrating it, processing it
Yeah.
in meaningful ways, arriving at a decision, and then, imagination and
Yeah.
creativity. And that's really hard because, we have 100 billion neurons,
Yeah.
100 trillion synapses. And in theory, to really understand that, you're going to have to know what's going on in many or maybe even most of those.
Yeah, that's the problem. It's a as I've said so often, that's why I do um uh why I do physics, because it's so much easier.
So have you secured a seat to Mars yet, or the moon?
No, I'm happy right here. I'm not gonna. I mean, I actually, the moon I would like to go to. Mars I have no interest in heading to, especially since I know it's a death sentence. But uh uh no, it's
Lawrence, whenever a lot of times at night, I lay in my bed with
yeah,
uh, a Kindle, and, I'll look at the latest James Webb images, and kind of lay there for a few minutes and, just kind of soak it in and think about how insignificant we are, but yet how amazing it is.
How amazing it is. Those are the things. And think about the brain and the universe together are the two things that make me realize how amazing and insignificant we are at the same time. But you just convinced it's a wonderful way to end, because it just occurred to me that the James Webb Space Telescope is good for health and brain function, because it causes us to lie in bed and think about things, exercise those neurons. And that's what's great about thinking and being a human and being inspired by science. And the science you're doing is inspiring. And I appreciate you taking the time to not just write the book, but spend time with me. It's been a lot of fun. And I look forward to more good things. So thanks again. It's really great to see you again. Thanks.
Okay, you're welcome, Lawrence. And thanks again to you.
I hope you enjoyed today's conversation. This podcast is produced by the Origins Project Foundation, a non-profit organization whose goal is to enrich your perspective of your place in the cosmos by providing access to the people who are driving the future of society in the 21st century, and to the ideas that are changing our understanding of ourselves and our world. To learn more, please visit originsprojectfoundation.org.